Thanks to everyone and welcome for attending this webinar of the UK CEiRSI, the UK Centre of Excellence on In-Silico Regulatory Science and Innovation. And this is a series of training and pilot webinars that are proudly supported by Innovate UK, the Medical Research Council, and the Medicines and Healthcare Products Regulatory Agency, MHRA. But they are made possible thanks to a lot of colleagues both in the UK and abroad that have engaged with the In-Silico regulatory Airlock initiative, of which this is one of our last pilots. We have two more after this one before coming to an end of this piloting phase. Our webinars will delve in the latest advances, best practices, and applications of in-silico approaches, fostering collaboration and knowledge sharing among stakeholders to drive progress in healthcare and medical research. And we do that through this process, which is a safe space for pre-competitive discussions, where all of the examples that we are exploring, we want to focus more on them as an exemplary role rather than on the particular brands or technologies involved. Without further ado, we will be putting a few resources on the chat as we go through the event, but I would like to hand over to Tim Denison, Professor Tim Denison from the University of Oxford, who will give us a framing of this Airlock on in-silico trials in deep brain stimulation. Thank you, Tim.
Thank you, Alex. And so we've put together kind of a high-level summary of our.
I'm sorry, I put it on the agenda because I was coming anyway.
Samuel Trupp. Okay, thank you. So the group with Tori Marks, we're going to start with an introduction to kind of frame up the problem, giving you an overview of a Parkinson's device, which is kind of embodies the principles of the PCLC, but also highlight some of the problems we're interested in exploring, and then queue it up within the context of the overall in-silico model framework that you might have seen in other webinars. Then we're going to cut over to two much more specific examples, one from Jeff Herron, who's worked to get an investigational trial going in the United States at the University of Washington using an investigational device, and that's been working with the FDA on some lessons learned there. Then we're going to come back to the UK, and John Fleming will be giving an overview of some algorithm evaluation he's been doing to support a trial in the UK with some similar themes that start to arise in spite of the different applications. And then we'll loop back and open it up also to Alec Widge and Tay Nedoff, who are also members of the core team, and discuss some of the higher level themes of the model verification and where we think we are today and where we'd like to go in the future. And then also Stephen will help to facilitate some follow-up on model integrity. So we'll get started with Tori and me. So go ahead, next slide. And first of all, we would just want to start with a high-level vision of what the team here is trying to achieve. And this is in the area of applying bioelectric devices in some way to restore a physiologic process. And so our model is invoking the good regulator theorem from back in the UK cybernetics work in the 70s, where a good regulator oftentimes takes on the attributes of the system it's trying to regulate. And so for us thinking about physiology, this invokes adding feedback mechanisms. So we're going to respond to immediate changes by measuring sensor data, having a control algorithm model that then reacts and adjusts the next action policy, but also at the same time thinking about rhythms such as circadian rhythms and how time needs to be factored in for prediction elements. And then our overall objective is to improve these active devices to, in the short term, restore physiological rhythms for therapy optimization and apply these algorithms for that purpose, but maybe in the future, use it to slow degeneration. It's a more aspirational topic. That said, we need to think about the challenges of building such a closed loop. So when we were getting started on these designs, a decade or so ago, kind of where we were was in these open loop devices, although you're going to hear from Tori, we don't like to call them open loop, but manual devices. And this is an example from a tremor patient where on the left, his device has been turned off, and on the right, his device is turned on. And so you can see that he is getting therapy benefit, but there's still some residual symptoms that are showing. And part of this is that we can't keep turning up the stimulation because just like a pharmaceutical, you'll start to invoke side effects. And so one of our objectives is to say, what are the degrees of freedom that we might apply to improve therapy, open up that therapeutic window for fuller symptom relief, but at the same time, avoid side effects. And as Tori is going to reinforce, tremor was relatively straightforward for us to go after, but as we go into newer areas such as epilepsy, and then stroke rehab, like Jeffrey's going to talk about, the challenges of doing an open loop manual device become fairly significant. So we need alternative technologies. So kind of how we got there was this notion of a reversible lesion. So the Benabide and other innovators were taking what historically was an electrode placed into the brain target, such as the thalamus shown here. And instead of applying an RF lesion, they would place an electrode that could then be tunneled up through this, once it's placed in the target, tunneled underneath the skin, and then hooked up to basically a modified cardiac pacemaker placed in the subclavicular area of the chest. And then a fixed pattern of stimulation was given pretty much 24/7. And that was the notion of the reversible lesion, is that you could adjust the stimulation parameters and use that to titrate the device on demand. But calling it a reversible lesion also anchored the field a bit in terms of this idea of open loop or non-responsive therapies. And so the trend in the last decade has been to say, can we expand beyond that and take advantage of modern electronics and modern controls?
So from 1990 till now, pretty much all of implanted neuromodulation stimulators have followed this like classical neuromodulation adjustment. And as Tim said, it's not really open loop. I like to think of it as closed loop, but it's a manual loop. So instead of feeding everything into a machine, all of the feedback stuff, all of the EEG, all of the physiological and behavioral factors feed back into the clinician to integrate and decide how to adjust the stimulation. You can imagine that this is a very slow time scale for update. It'll be every 1 to 3 months whenever a patient goes in to see their GP or their specialist. So humans are the algorithm that determine the policy to control the loop. This is a very slow way to adjust to symptoms. And while this was okay for something like tremor, which has a very obvious physical response that changes immediately after stimulation changes, this is going to be a lot harder with things like epilepsy, which has unpredictable timing and variable morphology of the signal of interest or the symptom of interest. And stroke rehabilitation will be even harder. How do you track plasticity, and what timescales do you even need to know? We're not sure yet. So as we've gone into 2025, we've tried to really make this loop more automated and bring the clinician more on top of the loop than within the loop. So in this case, all of the stuff, all of the rules and parameters for the algorithm are programmed by the clinician, but then the algorithm can run by itself. So then as physiological and behavioral feedback variables feed in, acute changes can be made on the timescale of seconds or even shorter. And also like feed forward variables can be added into this. So we can add time of day or postural changes, that gives us information about sleep and wake cycles, many things that could affect a symptom profile, and the algorithm can respond to those proactively as well. So it's correcting for expected changes and reacting to stimulation affected changes in a much faster timescale than previous policies. One other thing I want to flag before passing back to Tim is that especially when we're thinking about these automated systems, is we need to consider what things get flagged and logged for future clinician reference whenever the patient does go in to see their clinician, what things are, or what detected events need to result in a return to that open loop, manual loop stimulation, and what things could create an emergency stop where the system just stops stimulating. So
as we're kind of heading into this future state from the clinician in the loop, if it will, as a manual process to the automated loops, it really brings up some new questions and risks. And this starts to queue up the role of in-silico, or what we will call hybrid in-silico models. That'll become more clear as we go through our 2 use cases. And that includes understanding the nature of the input signals and how we can model those in response to it and how it interacts with the device, exploring biomarkers, considering artifacts that are potentially problematic. And we'll go through 2 cases of that where the algorithm might be confused and have different responses than what you desire. Thinking about the set points, actuators, limits, and also, as Tori was just saying, what are the fallback modes and how can we demonstrate those fallback modes? So the kind of our exemplar to set the stage is from the area of Parkinson's disease. This was a paper, came out 12 years ago, and it was, I'll walk you through it. Starting at the bottom left, the mental model of operation is that when you measure off of those electrodes that are placed inside of the brain, you can see these local field potentials. And if you look in a Parkinson's patient in the STN, if you do a spectral analysis around 20 hertz, you see that defined peak and the LFP power, and that correlates with the symptoms of the patient to first order. And so as that peak increases, symptoms would increase. As it goes away, symptoms would go down. Now, what's important is that we can measure that power. So we sense the sensor is the local field potential. The algorithm is to extract the power in that spectral envelope and compare it to a set point. And then just like your home thermostat, when we cross that level, it goes above it, we can gate the stimulation on as the actuator. And when then it falls below that level, we gate the stimulation off. And what was promising is that this adaptive approach was showing incremental benefit compared to continuous DBS, and certainly random stimulation, and also potentially saving some energy power. That was a promising experiment. That was a promising experiment.
Okay.
Yeah. Back one. That was a promising experiment in the laboratory, but getting it out of measured with externalized leads, getting that into a chronic situation where patients are sleeping, 24/7 operation, and lasting over several months to years required quite a few issues to be resolved. And that was motivated by our checklist. So let's systematically step through those real quick and then talk about where In-Silico can help. So first is, what's the nature of the signal that we're interested in? And so when we go and build a brain interface, and we'll have examples from each of these in our deep dives, we can think about an evoke signal. So we're going to actually stimulate the brain, say with the pulse, and measure its response. And that's shown as the P300 evoked potential as one classical example. And Jeffrey's going to walk through some of his work and looking at evoke potentials and how those can factor into a rehab. The counter or complementary approach is to look at these baseline rhythms. And so these are the inherent rhythms in the brain, like we just discussed in Parkinson's, in the 20 hertz range, the beta rhythm. And this is an example off the motor cortex, say around 10 hertz. So one of the real challenges in this space is that the signals are 1,000 times smaller than, say, a cardiac pacemaker. And so one of the things we really need to work on with a new device is, can we resolve these signals, especially in the presence of stimulation? So that gets into our next area of the biomarkers. And so interestingly, there's been studies where we see pharmaceutical responses, say, in the Parkinson's brain. That's on the left-hand side, where there's on versus off drug. And then that was replicated using stimulation as a complementary therapy. So in this case, there's the rhythm, the beta activity around just above 10 hertz. There's a staircase step of stimulation, and where the red bars occur, and you see the beta falling below a critical threshold, so symptoms were relieved. When stimulation was then turned off, the beta rebounded, and then turned on, it went away again. So this is an important part of developing the in-silico model, is trying to mimic this behavior in a way that we can then test devices out systematically. And one thing that's really critical, and we'll get into in the artifacts, is that the stimulation is invoking signals on the order of a volt, 100 millivolts to a volt in the neural tissue surrounding the electrodes, while these signals that we're trying to measure on the order of microvolts RMS. So we can have measurement requirements in the order of a ppm, part per million, and that can be a real challenge when we're trying to develop a new device. And so using the hybrid in-silico gives us a mechanism to actually test out the capability of any new device. So in terms of artifacts, one of the ways we can model the physiology and the device interaction is kind of the latent signals in the body. So one of the challenges that we've run into is that in the subclavicular implant, you can see we're putting the device right over the heart, and this can create a electrocardiogram. Oh, sorry, I'm just reading Steve's pop-up. So this can create an ECG artifact, which you can see peaks right around that 10 to 15 hertz band, right where our beta signal is of interest. So if any cardiac artifact bleeds into the measurement due to device issues, that can actually confuse the algorithm and actually artificially say continue to have stimulation on. And so that's one of the things we want to model in the hybrid system, is kind of create these artifacts and see how the device operates in its normal operating mode. Yep. So the key. So the other issue that we have to work about is, remember that the stimulation is coming through. So we have to think about the stim interactions. So just like your cell phone, where you have to transmit to the radio tower and then listen for what's coming from the tower, and those are orders and orders of magnitude different, we have the same bidirectional interface challenge in a brain interface. So if you can do down one, the first issue is we have to think about that interaction. So first up is, go build. We stimulate, and the problem is whenever you transition stimulation, it kicks the neural interface. And so you get this broadband jolt in the interface that then bleeds through the detector. And so then down, that then turns, so you turn the device off because the physiology said I no longer need stim. But when you turned off the stimulator, the physical interface, the tissue electrode interface, just, electrochemistry, glitches. That can confuse the algorithm. And it says, oh, wait, I need to turn stimulation back on. And so then as you go forward, say, oh, no, we didn't really need it. We can then turn stimulation off, but as you turn off, you glitch. And what this does is actually create a relaxation oscillator. And so if we're not careful, this bidirectional interface can actually decouple from physiology and be completely triggered by the nature of the device and the artifacts that it has with the tissue electrode interface. And so this is a key area for us in terms of mimicking the brain's behavior and putting it in a hybrid test environment and showing that the algorithms maintain stability and actually don't go into this oscillatory state. So another area where in-silico has its opportunity is looking at circadian patterns. One of the things that's been very interesting as we put these sensing devices out into the field is that we can measure physiological variables over very extended time frames on the order of weeks to months and even years now. And in this case, we're looking at that beta rhythm, but what we're doing is every 10 minutes, we're saving an average sample. And so as we look across the time axis on the bottom left panel, you see this very repetitive behavior, basically 7 dips over every week. And that's the beta rhythm falling as the patients go to sleep. And then it rises back up during the day. And so this is something else that we want to model, because of course, just the time when this gets into verification, validation of a device, we can accelerate these times and kind of create, accelerated rhythmic periods to help to compress time and show still that the algorithm, though, is behaving as it should, both during the awake state as well as during the night. And then there are other opportunities where there's rhythms, say in epilepsy, where we can actually show that the predictive elements of the device can be modeled and captured in a hybrid in silico environment. Another area, and one of our big opportunities, we'll only say partially achieved, is thinking about different mechanisms for algorithms. And so the example that was provided by Peter Brown at the beginning was that on-off switch, kind of a two-state controller for Parkinson's. An alternative approach was explored by Jeffrey, actually our next speaker, and Helen Bronte-Stewart at Stanford, where there was a two-state algorithm that actually looks a lot more like a homeostatic system, where when the beta crossed above a certain threshold, an upper limit, you would turn the stimulation and keep incrementing it. But then you'd enter this region of no change, and then kind of the physiology is allowed to wander, and then there's a lower limit, and once the spectral power fell below that limit, then you would start to back off of stimulation. And so in a recent study, these relative algorithms were compared against each other, and there were actually statistical and clinical differences that were observed. And so one of the big exciting areas we see in the future is, can we use in silico models to basically co-develop algorithms and test them before we bring them into the clinic? But that will require us to have a true model, kind of a true model of the brain's dynamics and how it is responding to stimulation. Then the last part is thinking about safety in systems. And so the examples I use with students is thinking about the human factors associated with automated systems like cruise control in your car, where there's clear understanding of how to disengage the loop and the action to take, tap the brakes, for instance, and pull out of the responsive mode. And then there's other cases such as the 737 MAX, and thinking about single point failures in sensors, human factors for training, and other kind of both full system analyses and how things can go wrong and the appropriate actions are taken. And one of our advantages in deep brain stimulation, at least the historical areas of Parkinson's and tremor, and a little bit with epilepsy, although it's still evolving, is that there is some marginal benefit for open loop. And so in terms of a fallback out of an adaptive mode, there's kind of a known condition. But in emerging areas like the one Jeffrey's going to talk about, that fallback mode is not necessarily as understood. So one thing we want to capture, and this is a wonderful case example from the FDA putting out some guidance. It's a guidance on the design of physiologic closed-loop control technology, really summarizing a standard that's out there, 60601-1-10, which is a very general purpose standard in terms of covering things from artificial pancreases to automated anesthesia, baby incubators, and the NICU and the like. But we also are starting to look at how we can apply this in the area of these, neuromodulation, bidirectional brain interfaces. And we think ultimately we can take this framework and combine it with some of the hybrid in silico models here and work towards a medical device development tool. And what we think is missing in the application of these standards is kind of a set protocol with pre-recorded signals and characteristics that any new device can go and be explored with. And so this is a schematic. We'll circulate a paper as a follow-up to this talk, summarizing the standards. But this really captures those risk considerations we were just talking about within each element of the automated loop, in terms of human factors, people having a mental model of operation, thinking about safe fallback modes. If for some reason our algorithm goes into an unknown state, does it actually go into a safe fallback in a known manner? That's something else we can test, by mimicking a behavior with an in silico model, testing our logs and our alerts, keeping track of the device operation, verifying the actuation limits, and also thinking about variations in patient dynamics. A key element in any feedback loop is what are the variations in terms of the plant dynamics. I don't mean literally a plant. I'm using control, of course, the patient's physiology dynamics, are there any delays? Are there any low pass filters? And taking those into consideration. And the benefit of the in-silico approach is that we can actually really perturb those and drive them to extremes, kind of take them, do a full exploration of the parameter space that might be a real challenge for us to achieve in any finite clinical data set. So this is just to summarize, like this Parkinson's example kind of highlighted many of the motivations for why we want to explore physiologic closed loops, but also some of the motivations for why we think in silico techniques can help us to do some bench verification. That certainly before we start a trial, we know the device is passes its risk-benefit premium to justify moving forward with the trial. And this, the device or the algorithm I was just talking about is now on the NHS list. You might have seen this in the spring. This is a patient actually up from Newcastle, with part of a group I collaborate with, Luke Bashford and his team, that was covered by the BBC. But looking forward, next slide is, this is just one example of this vision of us building up devices with predictive and reactive closed loops. But in order to move the field forward, we really think there's a role for hybrid in silico to help verify devices, a little bit of validation before we start the clinical trial, but ultimately perhaps use it for more exploratory validation of algorithms before we commit, and thereby accelerate our pathway of innovation. So our general purpose, or I'll say this, we do have an inspiration from diabetes, like I say in the PCLC. We just wanted to kind of plant this seed in people's heads, is that they're quite a bit ahead of us, and we can look to them for enlightenment. So they do have reasonable models of diabetes patients, the way that the pancreas responds to different challenges, glucose, carb loads, things like that. They can have a suite of different patient states and kind of get a population. And then in Simulink, go through and actually model and validate some of their algorithms before they commit to a clinical study. And so I'd say this is kind of our true north of what we look at, a little bit with jealousy. Say the pancreas is a little better understood right now than many of the brain circuits we're exploring, but it does give us a model of what we want to strive for. So now to set up Jeffrey and John, we're going to get the first part of this in-silico worksheet. And so to summarize, our goal is to develop systems for bidirectional testing of an adaptive brain interface, where we're going to explore these personalized closed loop therapies. But our first emphasis for where the field are now is on safety. So showing we can maintain stability and safety over a wide range of real world conditions, and that this helps to justify the regulatory approval for our device trials. And so what we're going to do is walk you through two use cases of our hybrid in silico, because this is where we're basically coming up with simple, some might even say toy models of the brain to get started. And then with that system in place, immersing a device into an environment that mimics the brain and then running it through its relative paces. In one case, for the application of evoked potentials and characterizing those for Jeffrey's work in stroke rehabilitation, and then in the area of seizures and interictal responses for an adaptive system for epilepsy with John. And so with that, Jeffrey, we'll actually stop sharing and see if you can run it off your computer.
Yeah.
If not, for a local. So you've got
should be able to. Can everyone see my screen?
Yes.
It's come up.
Wonderful. All right. Well, first off, by way of introduction, I'm Jeffrey Herron. I'm from the University of Washington. I'm an electrical engineer that got embedded within a neurological surgery department. Though, as full disclosure, I do have some experience in the medical device industry. I worked actually under Tim back when he was at Medtronic in his research and development organization. I'm currently at the University of Washington, which is out on the edge of the Pacific Coast, out in glorious Washington State. University of Washington is one of the larger public recipients of NIH funds. We have a large student body, and we've kind of established ourselves as one of the large public institutions that's in the United States that's really kind of deeply involved and invested in exploring clinical neural engineering and working to improve the outcomes of patients with different disorders. My work is largely a lot of system integration engineering to kind of support clinical research and clinical trials, emphasizing kind of near-term translational work and establishing feasibility of new device concepts. And really my focus is in this bidirectional neural interfaces, where we have neural interfaces that can both sense and stimulate to treat movement disorders like Tim just discussed in regards to Parkinson's, but I also do work in neuropsych and then also neurological injuries such as stroke. And today I'm going to be discussing some ongoing work that we've been doing related to the CEiRSI kind of objectives as a case study, focusing mostly on our stroke work. And stroke is, of course, one of the top 3 causes of human disability. I'm sorry for I don't have the UK numbers, but I'm sure this is a worldwide global problem. In the US, there's 600,000 first strokes every year, and the vast majority of these patients all have enduring difficulties with daily life, major quality of impact requiring large amounts of care and support for patients who have suffered from these disorders. So the primary means of treating stroke is still to date just primarily physical therapy. And this is really, at this point, not enough. Many patients do not recover even partially with rehabilitation. And at some level, there's this understanding that we're only engaging with the periphery, with the muscles, and that's only, in different ways, it's indirectly affecting the brain. And so our key driver of our work is really how are we engaging the brain, and we're choosing we're engaging the brain based on 4 key observations. The first observation is that stroke recovery of motor function is heavily influenced by surviving connectivity. There was about almost 10 years ago, there was published results from the EVEREST study, which was a North Star Neuro clinical trial to implant a device to deliver open loop stimulation to the motor cortex post-stroke. And sadly, the trial failed because they failed to meet a key endpoint at the 12-week mark, where they couldn't actually distinguish themselves at control. The tragic nature of the study, though, is that for all the patients that had proceeded past that, at the 24-week time point, there was a large amount of patients who had actually benefited. But in just the kind of cruel world sort of way, the failure of the 12-week endpoint was a kind of a critical milestone, which required the termination of the study. Now, when they were doing even further subgroup analyses, really to peel apart where were people doing well or where were people, failing, the surviving cortical spinal tract was identified as being the key driver, i.e., you can't really fix what's been completely destroyed. If there's actually, motor connections to the limb that are surviving it, patients actually did even far better than what the initial analyses were. So there's this first observation is that enhancing connectivity, really having connections and being able to enhance those connections, really might be key to improving rehabilitation outcomes. The second observation that really drives some of this work is that electrical stimulation can modulate neural connectivity. This might seem, perhaps obvious, but when you get into the actual neuroscience of how are you measuring cortical connectivity, one way of doing that is you can either do this from functional or structural manners. A functional way, i.e., measuring the actual information flow, is a cortical evoked potential. And I'll talk a little bit more about evoked potentials in a couple slides here, but this is one way to quantify cortical connectivity by basically more or less delivering some stimulation to the brain and see where you see responses. If anywhere you see a response, must be connected to in some foundational way to the site of stimulation. What's really exciting is there's been some really great clinical and animal work that's shown that if you stimulate in the right way, you can actually increase the responses. You can tune the gain of these evoked potentials in response to that stimulation. And one particular way of doing that is by delivering stimulation in time with the natural rhythms of the brain, right? And so if you stimulate in the trough of the beta rhythm in the motor cortex, for example, you can actually enhance the connectivity, increasing the size of these evoked potentials. So the key observation really here, though, boiled down to is that cortical stimulation delivered in an activity-dependent way can actually enhance connectivity. Third, enhancing connectivity is actually, from a clinical science kind of review perspective, will likely cause enhanced stroke rehabilitation outcomes. This is one which is looking at more than 200 publications. Dr. Kramer of UCLA, who's a co-PI in this project, has basically argues that there's overwhelming evidence that improving post-stroke functional connectivity will cause improvements in post-stroke recovery by looking at the causal relationship of connectivity on rehab outcomes. And so this was more or less performed through a large amount of imaging work, reviewing, what would be counted up to thousands of cases of different, MRI and DTI and connectivity measures that then, in looking at how outcomes actually were done. So through literature review, it's more or less established that there's, this overwhelming evidence that improving connectivity will help. And then finally, and importantly, the signals that we care about for all of these connectivity enhancement measures survive in stroke. We've done work ourselves, and others have also observed this, where the rhythms, the beta rhythms in the motor cortex, even in someone who has mass amounts of disability, we still see beta-related desynchronization to movement. And we also see cortical evoked potentials, which can be evoked and enhanced using phase-triggered stimulation. So what this is important is that everything that we've required in the kind of observations 1 and 2 actually are preserved in stroke as well. So put together, this is really the key hypothesis that drives our study design, and thus the engineering goals that we need to do to accomplish this is that we're expecting that, to improve post-stroke recovery by enhancing neural connectivity through the use of phase-triggered stimulation delivered alongside conventional physical therapy. So we're trying to enhance rehabilitation outcomes with a implanted device delivering stimulation to the brain. And so this is a NIH-funded clinical trial where we're going to be recruiting 12 stroke patients who have chronic deficits. So these are all subjects who will have already gone through some amount of completed their normal physical therapy and still have deficits. So these are people who have plateaued. It's been at least post 6 months from stroke. The project has been going a little bit for a little while now. Excitingly, we had our first patient implanted in July 2025, and we're proceeding. The device we're using in this work is the Cortec Brain Interchange. Cortec is a company in Freiburg, Germany. They make a 32 contact ECOG focused device with sense and stimulations shown over here. The device can do bipolar stimulation with any combination of contacts, and it can stream a kilohertz of ECOG data from each of the channels simultaneously. It works with a PC in the loop, so there's an external unit that basically handles the transfer of information flow to the computer, where an application is running that then performs any sort of processing we want to do to then trigger when and how stimulation is delivered through a command back through the external unit. This is actually a reconstruction of the device from our implanted in our first patient, along with the electrode localizations. So you can see that basically with these with our electrodes that we have, we span the parietal, sensory, and motor, and into premotor areas to basically look at the full kind of connectome, so to speak, of the motor focused regions of the cortex. And we do this with 2x8 grids that were both basically implanted during a craniotomy, which you can see the cut marks in the skull in this one. Okay, so let's go back to the motivating hypothesis. I'm the engineer in the study. What is it that I need to do, right? The first one is we must demonstrate the capability to accurately record and measure CCPs to evaluate our capacity to induce neuroplasticity, right? We can't, if we're going to make the argument that we're going to be enhancing neural connectivity, we need our device to be able to actually measure that induced neuroplasticity. So both Tim and I have been saying repeatedly that we were going to get into evoked potentials. Here's the time when we're finally doing it. Evoked potentials can, I think, most be easily considered a type of brain radar or sonar Tim will probably get a kick out of that because we've been talking about brain radar for a very long time. But basically, you're giving electrical pulse to the brain, kind of a ping, and then you're basically measuring at where you see responses back from the circuit. And the magnitude of those responses gives you a measure of just how much kind of information flow there is between those different areas. So this is a functional connectivity in terms it tells you how brain regions are functionally connected. And this is kind of in contrast to a structural or more like an anatomical measure of connectivity, which might use a DTI and look at just where tracks are going. This gives you more of a measure of how are those, what's the gain on those tracks that are existing? You know, how much does the receiving side and the transmitting side, how much does the receiving side pay attention to a specific area? So that's why we call this a functional measure of connectivity. There's a lot of examples of evoked potentials being used these days in different types of work. One area of use is they're being explored as a way of evaluating critical pathways actually as kind of like something to do during DBS implantation for a DBS for Parkinson's disease. Placing a cortical strip temporarily on top of the motor cortex, when you have it can be used as a sensor for an evoked response from the depth probe, which kind of indicates that you may be in the right spot. But one thing I want to point out here is in this prior work is when you're delivering electrical stimulation, you know, to the brain, the amplifiers used in the actual devices to measure these sort of systems are, of course, you know, operating in very different, you know, amplitude domains to sense neural activity. So it's very, very common for you to have fairly large stimulation artifact that kind of, without, you know, doing too much characterization, can be difficult to pull apart from your evoked response. So what's sometimes used is a reversing of polarities or stimulating and sometimes doing sonar pings twice, one kind of a more positive oriented pulse and one being a negative oriented pulse. And you would expect a stimulation artifact then to kind of flip polarity as we see here, whereas an evoked response would be, you know, the actual deflections of the signal itself would be preserved kind of regardless of what directionality the stimulation hardware was delivered in. So at some point, though, the key thing that with any of this, though, is we're measuring signals in the presence of a lot of noise, right? And so the neural signals underlying in the neural rhythms, it's not just necessarily 60 hertz. We have, you know, also at some point, the brain is never not active. And so deconvolving the time-locked neural signal recorded with stimulation requires performing a large number of trials and then kind of averaging the results to pull out an averaged evoked response. So these are not single sonar pings. These are you doing 50, 100 sonar pings and then looking at the average response, and that gives you your measure. And then once you have that active measure, you can start to pull apart different components, which are often looked at in relation to some zero mean baseline, where there are negative deflections and positive deflections that all have average amplitudes and average latencies in kind of respect to the stimulation pulse. So now, while that's evoked potentials in the idealized form, there's a lot of practical measures that actually make this very difficult to do. And our group has spent much of the last, a lot of the time in the last decade recording evoked potentials during both DBS surgery and in epilepsy monitoring. And the confound is this: when you don't see an evoked response potential, there's this underlying uncertainty. Is the sensing configuration influencing whether or not the evoked potential is working? Is the stimulation artifact masking the evoked potential, right? Or is there a problem with the data processing, or is there just no evoked potential at all? And then when you do see an evoked potential, you have to double check and do a lot of work to make sure it's not an artifact. And so the key reason why this is a problem is that there isn't really any independent way of measuring the ground truth in terms of working with an actual patient that will give you, confidence necessarily without a whole lot of exhaustive work to gain basically confidence in this. And this is because in large part, the systems are highly susceptible to the configurations of the neural systems that we're using to interface with them. So this is some work that was largely led by my former PhD student, Dr. Hanbin Cho, who recently defended her thesis, but she's now a postdoc in the lab, along with Professor Tim Dennison and Moad Beneger, both from University of Oxford. And we were working on methods to solve this problem through a saline tank model test where we use an active piece of equipment that's sensing. So it's not just enough to basically have our system stimulate into the saline tank and then measure the response. We need our test rig to pay attention to what the system under test is doing and simulate. In order to simulate and evoke potential, it needs to know when stimulation's happening. So we're turning our test rig into a closed loop system itself, which is trying to react in response to what the system under test is doing. And this is what makes this actually, from a electrophysiological design perspective, a system that really opens up a lot of exciting opportunities to characterize a system's capacity to stimulate and evoke potential. And so, this is an example of what we can do in this sort of saline tank. We have when the system is just rendering an evoked response, we have the simulated evoked response here. When the device under test, the cortex brain interchange is stimulating, this is the more or less the stimulation artifact. We have a long return to baseline, and then we can of course layer these two tops on them by enabling this kind of closed loop test rig of sort of functionality where we're having the stimulation evoked response evoked when the brain interchange performs a simulation pulse, and that lets us then now go at our basically look at, for the first time really with a known ground truth, how is our system actually performing in terms of sensing and evoke potential? Are there, in some ways, are there different components that are being, either corrupted or difficult to see? And our latency is being adjusted. And how repeatable are these measures? How many tests do we need? These are all things that are now finally characterizable with a platform like this. And this really allows us to then, also test all the various simulation configurations that we can use and look at on the, trial by trial basis, what is the system doing and how is it sensing that evoked response? We can also then use this as a key part of our signal analysis pipeline. So this isn't just supporting kind of live interactions with the device, but through collecting this data, we can then, using this established known truth, we can start to then pull apart how do we and can we, in a post-hoc process, analyze the data in a way that we can actually now extract this. And with this particular test bed in the brain interchange, we found that we could use ICA and some curve fitting approaches to basically more or less pull out the original evoked response from the frankly very messy raw data. So study engineering needs two. We need to deliver phase triggered simulation in a robust manner, in a closed loop way, in order for us to actually be able to induce this neuroplasticity. And so this is the second need that we need. At some level, when you're building a closed loop system, it's very easy to start out with kind of doing feasibility testings with established kind of benchtop equipment, oscilloscopes and function generators. And we can use these sort of means of establishing feasibility, i.e., if we have a perfect sinusoid wave, it will the system encoding latencies be such that it's even possible to deliver stimulation at a specific phase of beta, and we can do that. But at some level, we start to see, immediate limitations in that, we have ringing of our filters because we're putting a 0 to 1 sinusoid wave into the system in a very non-physiological way. And really, at some level, you don't really have a good sense of just doing this sort of approach of how the system works with actual physiological interface. And so at some level, this, saline tank methods doing neural playback has been a key part of kind of establishing actual functionality and, and moving beyond just a static feasibility testing rig where we could just time our simulation right based on the measured beta wave, required us to implement a more sophisticated phase locking methods in our research application, and we could do a test with actual neural signals that we collected in the with epilepsy patients to monitor can we deliver phase extim at specific phases of beta. And at first glance, it looks like yes. Look at the we're delivering stimulation. These are stimulation pulses delivered at in the depolarizing period, and this all looks good until we look a little bit closer, and we notice that the blue and the orange traces here are a trial run with this is the raw ephys data without any stimulation being delivered, and in this case here, the raw physiology we're delivering in the tank actually didn't meet the threshold for when stimulation should be delivered. Instead, the interactions of the stimulation creating basically more or less adding additional power in our beta band, seen as increased deflections in our filtered signal here, triggered additional stimulation pulses being delivered to the patient at a time when it wasn't necessarily appropriate from the neural signals alone. So the system's actually triggering based on the artifact, not the underlying patient's beta. And at some level in our risk assessment approved under with the FDA, it's not a problem if we're doing more. There's not a safety issue if we are doing extra stimulation pulses than necessary, and that's because we're comparing ourselves to a 50 hertz open loop baseline from the prior EVERST study. But from a scientific and a clinical study perspective, if we're doing stimulation pulses when we don't think they should be delivered, that might inhibit the neuroplasticity and then affect our outcomes of the scientific and clinical outcomes of exploring that facet of this work. And so we had to then, of course, use this platform to introduce and test and characterize mitigation methods to basically evaluate how we could actually get around this issue and preserve our ability to stimulate in an appropriate way. And so there was, we implemented kind of artifact blocking methods to stop our filters from getting contaminated with the stimulation artifacts. And we also had fail safes where we would stop ourselves if we stimulated more than 5 beta phases in a row. We would give the system a break to basically make sure that we weren't just in a continuous ringing sort of situation. And with this in saline, we could actually achieve high degrees of specificity of delivering stimulation at targeted waves. Our target phase here was 210 degrees. And then to cap it all off, we then went back to the epilepsy monitoring unit where we collected a lot of data we were feeding into the tank. And in 2 different subjects, we tried the phase-specific algorithm with this unimplanted benchtop to basically test under real physiological conditions where the brain would actually respond to what we're doing, not just simply in an artifact way, but also in a physiological way. And we could show that we could also still maintain similar performance to the saline tank, where we're delivering stimulation during the targeted region as desired. So I've already touched on one key need here, which is really modeling the response. At some point in both the evoked potential work and in the closed loop work, we're using kind of static replays of neural data, and we have to go back to the human patient in this way to kind of look at how confirm that the system is robust in throughout in physiological ways as well. And that's because the neural system is using simulated biomarkers which aren't responding to the changing stimulation or time varying parameters. And really, this is where I'm going to pass the everything back to Tim where we're going to continue talking about future needs, where we're going to need to embed models of the brain to test more sophisticated algorithms.
Great. Thank you, Jeffrey. So that was one example for stroke rehab with evoked potentials. And then John's going to look at the other complementary approach of looking at brain rhythms and adaptive methods for basically a trial that's been a device trial that's just been approved in the UK for treating pediatric epilepsy. Take it away, John.
Hi, everyone. I'm John. I'm a postdoc in Tim's lab working with Tim and Tori on this sort of in silico work. And so, yeah, just some disclosures. The device that I'll be discussing here today is just for investigational use only, and we're using it in a few UK regulatory approved clinical trials in the UK. So the trial I'll be talking about today is the CADET trial. So the CADET trial is a pilot study of open loop DBS, essentially for children with Lennox-Gastaut syndrome, which is a severe epilepsy syndrome. During this pilot study, 26 children are being implanted and will be receiving continuous stimulation to their central median nucleus of the thalamus. So historically, or to where we are now, is we've actually had 3 patients who've been implanted with the device and been receiving open loop therapy so far to try and help treat the symptoms of their epilepsy. But as a follow-on sub-study, we have approval for waking up the more advanced capabilities of the device, where we'd start to look at trying to more personalize the therapy that we're delivering, in this way doing more like closed loop based approaches. So just to give some an idea behind the mental model of how adaptive brain stimulation is working for epilepsy, before the patients had entered the CADET trial, and we can think of it as their brain has on this sort of y-axis graphic we have. On the y-axis, we're kind of plotting the brain's excitability. And then what's happening is when their brain excitability goes above a certain threshold, this corresponds to when a seizure might occur for them. So in this case, what we're showing is that in 2 areas, seizures have occurred as the brain excitability has crossed the threshold. When they've entered the CADET trial and started receiving continuous open loop DBS therapy, the thought is that this is sort of like reducing lowering the brain's excitability, and in this case, hopefully leading to some sort of therapeutic benefit where they're having less seizures. However, we still get breakthrough seizure events for the patients where seizures are still occurring, like they may reduce, but they are still happening. And so as part of this, we're looking to the next generation of algorithm where how can we improve upon this therapy and more so than what we're getting from the open loop results. And we think this is possible potentially with delivering closed loop stimulation. In the closed loop paradigm, how this would work is that the device will actually be continuously recording their brain activity. And when we detect of a seizure based on the signals that we're recording from their brain, we'll then be able to change or vary the stimulation that we are delivering to them at that time, hopefully to reduce the severity of the seizure or the duration of it. So in order to do this successfully, though, requires us to meet 2 kind of key technology requirements. First, as Tim and Tori talked to earlier in the talk, one of the risks is that when we are delivering these sort of these adaptive therapies, changing of the stimulation frequency stimulation parameters that we're delivering, we don't want to lock up the algorithm. Say if like changing the amplitude, as Tim had highlighted, can cause the system to lock up by essentially just delivering the boosted algorithm stimulation parameters. That may not be desirable, and maybe potentially in the long term, like worsen the outcomes if they're getting from their open loop therapy. But also we need to also ensure that we're able to maintain a good sensing and stimulation requirements in order to deliver the closed loop therapy. To do this, we've used a benchtop, which is similar to the system that Jeffrey has just spoken about. So in this case, we actually have the active implantable PicoStim device sitting in our saline tank environment. So the PicoStim Dynamo is the device that is also implanted in the children who are enrolled in the trial. The saline tank environment, as Jeffrey had mentioned, allows us to drive the field potentials across the tank. So, and then we can have the active implant, or we can then essentially simulate passing known brain signals or simulated signals with computational models through the tank, and then see how the active implantable responds when it detects these sorts of events for simulating seizures. The active implantable is also recording in real time, but then we can also using this neuro test bench, also record then the field potential that's happening in the tank. So it's kind of our ground truth. So for the epilepsy work that we've been doing, so as we move into this area of trying to work on the development of these closed loop algorithms for these patients, we've been using historical data that we've recorded from collaborators at King's College London, where we have one patient with a generalized epilepsy, where we have electrical activity recorded from their brain. And this was using a different device, which was an externalized DBS system where they were essentially able to record for a continuous 4-day period, where they actually had recorded 64 seizure events over that 4-day period. And in the cadet trial itself, we have also recorded data from one of the patients enrolled in the trial where they actually had a seizure while we were streaming from the device. We also have different sort of, we've also characterized, say, different activities from the brain, so kind of what their baseline daytime and nighttime interictal activity is like. So for this, for the testing methodology that we did, the first thing we looked at was then identifying what frequency bands are necessary for us to perform successful or good seizure detection. Then when we have detected a seizure, are we able to then respond to the seizure? So first with no stimulation, so is the device able to detect that it has occurred? Then test 2 that we did was, okay, based on when the stimulation is being delivered at one frequency and we detect a seizure, if we switch the stimulation frequency, does this impair the device's ability to detect or go into like a lockup state? So the first part of our results here were when we looked across the seizures that were recorded from the patient data set where they had the 64 seizures, we found that we were getting a pretty good separation. Well, during seizures, there's a pretty broad increase in power across the frequency content in the LFPs from the central median thalamus. So in this case, on the left, we're just showing that like in black, we have the baseline activity, so the non-seizure activity recorded from the patient. And then in the red is the LFP power spectral density of when the seizure, of the period when a seizure was occurring. We've kind of just taken here 10 to 20 hertz as a representative band for using our testing later on. On the right-hand side, we're also just showing out of interest highlighting the dynamic nature of the brain signal. So the interictal activity, sorry, on the right-hand side, we're showing the interictal activity for different portions of the day. So like a nighttime is given in blue, while the daytime activity is shown in black. What we can see is that the baselines of the signals, of the recorded signal itself, is actually varying depending on the time of day. But for the purposes of this, we're just looking at when seizures are occurring. So in our first test, using the saline tank, we essentially took the patient with the 64 seizures. We took their data and we had essentially concatenated the segments, snippets around when the seizures were occurring, and concatenated these sort of segments together into like just a continuous time stream where we have known times when seizures are occurring in the data set. We then pass this through the filters on the device. So we set up the dynamo, the picostim dynamo to be running with a 10 to 20 hertz bandpass filter. And then using this, we were extracting the power within this band. And what we can see is that, so in the bottom panel, we're seeing just the spectrogram of the data as it's being played in the tank. So during the time periods when a seizure was played into the tank, we can see that we get this like increased or broadband increase in power from quite low frequency up to almost about 50 hertz. The 50 hertz component across the spectrogram corresponds to line noise, as well as the saline tank and your test bench board are hooked up to a mains power source. So in this case, when we looked at the accuracy of our classifier that's running on the dynamo, we were able to get a very good area under the receiver operating curve of about 99%. So this means that we have good classifier performance at detecting the periods when seizures are occurring. So for the second test, what we wanted to see was now as we introduce stimulation into this system, did this then decrease the area under the receiver operating curve? Did this worsen the performance of our classifier detecting the seizures? So in the spectrogram now, what you're seeing is that in the periods where it's not seizures, what's happening is we're delivering a 6 Hertz stimulation frequency. So this is sort of like the background stimulation that's always being delivered. When the device detects in the saline tank that a seizure has occurred, it then switches to a high frequency, 120 Hertz stimulation frequency rather than the low frequency it was previously delivering. And when the seizure finishes, what we expect is that the device, if performing correctly, should then revert back to its baseline 6 Hertz stimulation. If we were to get algorithm lockup during this time, what we would expect is that when the first seizure occurs, the device would enter into delivering high frequency stimulation. And when the seizure would finish, that would then make the device try to revert back to baseline 6 Hertz stimulation. But because of algorithm lockup, it would then actually just see the change in the stimulation as being like that it needs to re-deliver the high frequency stimulation. The main point of note here is that we actually didn't see a huge, thanks to testing in this environment, we were able to see like, did we get a drop in the classifier performance when we were using these test signals? So we dropped from about a 99% area, 99% area under the receiver operating curve down to 92%. But this still is like sufficient for our requirements here, showing that we're still getting good sensing performance when we're detecting seizures and delivering low and high frequency stimulation. So we're not getting any algorithm lockup. So again, just to kind of go back to some of the limitations, which I think are in line with some of the points that Jeffrey had mentioned. In this case, we were replaying historical data sets into the test bench, which doesn't actually account for how it would be in, say, like a real patient, or where you would have different dynamical responses, or the brain would have a different dynamical response due to the intervention we're delivering. Computational models can be integrated into the neurotest bench. And this would then allow us to do more like dynamic sort of responses. So as we change the stimulation, can we change then how the signals are being driven into the tank as part of our test rig? However, current sort of simplified dynamics of the brain models that we've been using for testing kind of limit their suitability for clinical translation. And so that's kind of where we are at the moment. But the goal is in the future to have a sort of integration of these suites of models to allow us to more rigorously test the devices going forwards.
All right, thanks, John. So now we'll just do a quick summary, then we have Stephen come in and help to moderate the Q&A. So we'll get this slide back up.
The slide number for
Yeah. So going back, the high level motivation for multiple applications that we're seeing, from adaptive therapies for Parkinson's, epilepsy, using brain rhythms, but also evoked potentials, is the need for a kind of and right now a hybrid bench, but a way to verify basic safety of the adaptive algorithm in the present kind of a representative environment of physiology. At the moment right now, and so that's kind of our question of interest, is how do we develop such a test platform? And so we lost our risk. So is it not showing up, or?
I don't know what's happened to it.
Oh, okay. Well, so what we were doing is, when we think about the context of use, it's, putting together this hybrid system. We're currently not comfortable with fully modeling the tissue electrode interface because of some of its nonlinear dynamics. And so that is modeled with a representative electrochemical environment with the electrodes under test and a mix-up of solutions that try to mimic the saline environment of the body. And then we can inject signals in. And what you see in the baseline for the credibility summary is we kind of think about it as two stages. The first stage is just the core sense stimulation interaction and showing that the device behaves well, that under stimulation, the sense isn't completely corrupted, or we understand the limitations of that sensing. And so we can truly have comfort that in the extreme cases that we might run into in a patient, that the device itself won't have unintended side effects, such as the algorithm locking up on itself and going into an unknown state. When we think about the combination of this test model and the applications, what we put as the application risk, we still put as kind of in the medium level. Now, part of that we want to emphasize is because we use other degrees of freedom from the 60601-1-10 standard, such as limiting the parameter space of stim that's available to us. So you don't allow the stimulator just to go to any set level. We kind of bound it into a safe operating zone with the clinicians setting up those parameters that are considered safe. So that kind of bounds the risk, but we are relying quite heavily in terms of the actual model risk itself, because a lot of our release procedures, both in terms of the device itself, there we go, the device itself and the way it behaves under typical sense-stim interactions, is characterized and relies on this test. And then also one of the gating elements that we've at least done in our trial is we take the patient-specific records that we've recorded and the test algorithm that we want to explore, and we actually try it out in this environment and demonstrate that it will be well behaved and well characterized before we then set those parameters up into a patient. So that's why we ended up with the model influence. So the model influence being quite high because we are very reliant on it as a gating mechanism. And so that credibility summary in the short term, we have confidence in this neurotest DAC and the system and having it mimic kind of the real world and some worst case influencers that we might see in patients. But what we also want to look to is in the future, doing a better job of mimicking the brain. It's like that simple example John was showing. We're kind of forcing dynamics in our neurotest. What we'd like to get is to a model of In-Silico where it's actually modeling how the brain truly would respond. And similar to what Jeffrey was saying, can you come up with a model that truly captures the brain's plasticity networks and how it's going to respond to STEM, so we can get a much better sense of the capabilities of devices and refine the algorithms on the bench and in the In-Silico environment instead of relying so much right now on clinical neuroscience. So the outcomes, a couple things from this project. We do have a paper that Jeffrey's team and Alec Widge are championing and about to get out, and we would like that to drive its way towards a kind of loose standard. We're not ready to say it's a standard, but to actually start to think about a standard of how you get a de novo device. How do we actually create a well-characterized hybrid In-Silico model and demonstrate the safety of the device and its capabilities and benchmark it against other designs? And the other element towards socio-technical is us getting into it is thinking about really capturing and creating a library of patient responses across the spectrum and trying to build up and really understand the variants that we see. We thought a good closing slide and handing off to the Q&A as we've thought about the credibility evidence. So for us, the code verification, that kind of as device designers and 62304 and the like, we're comfortable with that. And the calibration, as you heard from us today, we're relying on pre-recorded signals from our representative patient populations, using that in a bench validation and getting towards, we'll say, in vivo as to do the crossover comparison of managing our hybrid In-Silico environment with what we see in patients. And that includes the populations as we build out our patient cohorts. Now, that's where we draw a line, say, for where we are today. Looking to the future, where we'd really like to get and kind of drive the field is thinking about emergent models. So can we come up with things that are actually predictive of how algorithms will behave in the real world and use that to build up greater plausibility? And that will be, that will increase the value of in-silico models by a factor of 10. Not that they're not useful today. They're very good as a baseline safety check, but in terms of truly accelerating the field and discovery, that's where we think we want to go in the future. So with that, that's our background of kind of the things we've been working on developing this space. We understand it's not the classic in-silico that you might have seen from some of the other ones. That's why we say hybrid in-silico. But we hope it does give you a sense of how excited we are about the space and where we think it needs to go. All right, Alex and Stephen, we can hand it over to you.
Yeah, it sounds great. I really appreciate your point here with respect to it being a hybrid in-silico. I think that I'd like to dig into that a little bit more just in terms of where you think the bounds are on that and how that relates to some of the other things that you've seen. I think I can probably speak for anybody who's been involved in the other pilots in terms of, this is all very interesting, and I still feel like, and you did an excellent job explaining it, but still feel like it's a bit over my head with respect to exactly some of the details of the modeling, but very interesting, and I appreciate the point with respect to the test bed and putting the test bed together, the value of that. I think what I want to do here is jump into some questions, and particularly I want to start with Joyce's question because I think it's nice and broad as it relates to the question of regulatory development. Now, I understand the test bed that you're putting together. You've taken some steps, maybe many steps, to move that into an FDA regulated either regulatory science tool or medical device development tool, something along those lines. So I think you've got some really good experience, hopefully, to speak about there. But Joyce's question that she put into the chat here is related to the different standards and regulations is appropriate for, and I think this is the crux of the hybrid situation. But as you think about the regulatory requirements for getting the test bed approved and in use, and then whatever associated regulatory requirements there would be around the algorithms that are actually in the devices. Can you share your thoughts or your perspective on the approval process for those two, as they are, I would imagine, intended to be separate, and then what the interaction is between the different models in terms of any credibility assessments and validation activities you'd need to do in both directions, from the testbed perspective in terms of, okay, well, what do you need to know? How well do you need to characterize the devices that you're going to use in the testbed? And then for the devices, how well and what are the interplays between the features of the testbed that go into assessing a device using the testbed?
Yeah, but Alec, can I put you on the spot a little bit? Because part of his Alec's been part of our community, and we haven't heard from Alec yet. So we'll let him go first, and then we can all chime in.
So I think in many cases, the phrase, and I think this is very appropriate because we hear it more in the UK than the US, is the concept of fit for purpose. Because so much of what is happening in this space is a device exists. Right? So the question about characterization of the device, in many cases, a device exists and is on label or validated for an existing use in the human body. And so from is this object safe to surgically put into a human, and will it basically behave itself what you tell it? That is no. But then there is a question, which is that device contains a certain number of capabilities, whether that's sensing, onboard compute, or therapy delivery. And the question is, does that set of capabilities, when matched against a reasonably expected envelope of neural signals, and as we've highlighted, dynamism in those neural signals, either wanted or just kind of happens, is the interaction between those well behaved enough that it stays within reasonable boundaries of safety and predictability. That is, in many cases, and again, I'm talking directly from what I personally care to do, and what Jeff described as an example of the sorts of things I also am interested in is I want to try to do a therapeutic mechanism that's never been done before in some number, let's say 10 patients. I believe that device A that is known to be safe to use in the brain could implement that therapy. But before I ask 10 people to let my neurosurgeon drill a hole in their head, I would like to have credible evidence that it probably can at least do it, right? It can deliver the dose of what I want. That's what a tool like this really enables, is it shows that there's ethical justification that you can actually feasibly carry out the study, which absent a tool like this is almost impossible. The second thing is you've heard in all of these talks repeatedly this concept of, well, and then when we apply therapy, the brain is going to change, either because, as in Jeff's use case, plasticity is the point, or even within epilepsy, it's well understood that once you start doing a closed-loop epilepsy therapy, you can change seizure frequency and seizure dynamics. You may change other aspects of neurophysiology. And in many cases, what the brain's going to do is unknown, right? We don't have data you can replay because it's never been attempted. There's no animal model, but you can come up with educated guesswork. You can say reasonably, it would probably be no more than this, no less than this. And with a good simulation test bench, you can then model out those edge cases and say, all right, assuming these physiological assumptions are reasonable, here is a couple paragraphs about why we think they are. This thing will be well behaved and safe. That is, from my perspective, that's very powerful because that's therapy enabling and sort of novel concept enabling.
And I think that's great. Thanks, Alec. And that's on. So I think one thing, Alec, is just a quick one to introduce yourself, because you're unique on our group. You're also a clinician.
Oh, yes, right.
Well.
I thought you'd introduce. So, yes, I'm a clinical psychiatrist, biomedical engineer, neuroscientist, and person who actively puts DBS devices into patients for both approved and unapproved therapeutic purposes. I am both the sort of intended audience of a system like this, as well as person working with Tim and Jeff to develop it, because it solves a problem I haven't been able to solve the other way.
So I thought that was important. So one thing I'll build on Joyce is really down to brass tacks as well. Is one of the early areas that we think would be promising is, Alec has these, armamentarium of tools available to him. But one of the things that we struggle with is it's a new enough field that there aren't really standards set, specifications of your sense amplifier must do this, that, and the other, and have this characteristic in response to stimulation. So one of our, it's not that exciting, but it's very practical. As one of the things we're first thinking about is an MDDT-like application, or getting into an ISO standard in the back. Like when we have a spec, we'll say this, for you to meet this standard, you must have this amount of stimulation rejection in these frequency bands. And then in the back, in the appendices, we actually have the test rigs and the setup and our models and a suite of models. And then what you do is you actually would just like we go to, Neurofens or any other, to do EMC testing today, this would be a similar kind of turn the key test, and we get the results back, and you either report on your results for your specification and how you're doing, or a pass, no pass, just to get started. Because what we're finding is the manufacturers like to highlight the good parts of their device and maybe downplay the bad parts of the device, and imagine that. And so we're trying to say, here is more of, from the community of users working on algorithms and trying to develop these therapies, here's kind of some baseline tests that pretty much any device should be characterized on. And then we can actually, for the Alex of the world and the clinicians who we serve here, in the UK, they can then make, informed decisions, as Alec was saying, and regulators like MHRA, when they're doing the risk benefit assessment as well, they'll actually be able to make an informed kind of data-based assessment.
I also
so Joyce, that's
really
what
quick,
I'm seeing, is kind of supplementing some of the standards as they exist as we move into this new area.
And importantly, at some level, just to riff off that for a quick second here, because I know there's more questions, providing industry members something to then design against is a key part of that. At some point, without having kind of design inputs, clear, justifications of like what's good enough on stim artifact rejection and things like that, they're kind of designing in a vacuum as well. So this is where we need to help them build things for us to use.
Yeah, on the funny side, many of our neuroscience colleagues, and even it was in the paper I showed, they show data and it says AU, arbitrary units. And then, well, what does that mean in terms of device specifications? And so for us, actually taking AUs and actually creating them into true specifications is a short-term objective.
Yeah, no, that's fantastic. And it sounds very, very useful. Love that. Alex, you have your hand up. What do you got?
Yeah, so this is really exciting, Tim and Tim. I think it's, let me play it back to you and think that this is in many ways a very different type of device and technology that we've been looking at. So most of these questions are guided to trying to understand and learn rather than necessarily disagreeing on anything you've said. But if I understand correctly, these tests could become something equivalent to the sort of 3-point test in the case of stents. So something that actually gives you a standard reference for measuring, that you do that for every device and give you sort of some basic characteristic. Doesn't, tell you specifically about the, necessarily about performance or safety per se, but it gives you something that is comparable between different devices to know how they benchmark to each other. So that's kind of my understanding. Could you explain a bit more for those of us who are not so much into neurotechnologies like you guys are? What is the current practice? Because in a sense, at some level, what you're proposing seems absolutely logical. And the question is, if that is not what is being done at the moment, what is being done? What is the alternative, right, that people use at the minute?
Alex laughing, so why don't we let him go first?
An ad hoc muddle of whatever you can cobble together on the benchtop in your laboratory, whatever you can con the device manufacturer into disclosing without your contact getting in too much trouble, and then whatever argument you can make to the relevant notified body. Right? There is no standard practice in this area, and I am not kidding about, at least within the scope of the US FDA, it is you make your argument and roll the dice on how the reviewer is feeling that day. Jeff and I were joking. Jeff and I happened to be at a conference 2 days ago, and we're only slightly joking about role-playing games and how you roll one 20-sided die, and if your number is high enough, you successfully convinced the person on the other end to let you do what you wanted. It really feels like I'm doing that much of the time.
Well, on that. Yeah, go ahead.
And Alec, just one point. This is really interesting, and I heard that you were sort of writing up some of these ideas, and maybe as a suggestion for your write-up. Oftentimes there is a lot of emphasis as if modeling and simulations are of a lower quality evidence than other things, right? Experiments, clinical, whatever. And I think we have on the line in the team as well, Amin Rostami, who comes from, PKPV and pharma. And one of the things he tends to emphasize is that in that domain, many of the modeling and simulation uses were driven by the fact that there was no way to do the experiment in the first place. So you're not trying to compare any gold standard that doesn't exist, but you're trying to provide basically to cover an evidential gap, a void, with something better than just the dice rolling that you were explaining, Alec. Maybe we need to emphasize more. We need to emphasize more to regulators and regulatory affairs colleagues that that's the nature of it, and also how the credibility of this approach or modeling compares to the sort of experimental modeling, i.e., benchtop and so on.
Yeah. So the I think one of the elements, part of it is it's a new field. I don't want to over-egg it, but it's so I think also there's a conversation of helping to show, bring people on the journey. And that's why Zach from FDA was on earlier, and they've been working with us on how we apply this physiologic control standards, originally designed for things that were not bidirectional interfaces or closed loop neuro, to this space. And so I think part of us is to gather and educate the regulators, also frankly, educate ourselves on some best practices. But I think what we find what's useful with it is actually a bit, Alex, to your point, like what we'll do is we'll intentionally imbalance electrodes. So we start to bound the design space. And what's nice about these hybrid in-silicos is we can make enforce failure. So like John and his epilepsy, he can adjust the amplitude of that seizure and make it go, when does it run into trouble? When do we fall below the signal noise ratio? And how does that compare to a patient population? The other one is intentionally imbalancing electrodes. So we completely mismatch the impedance, even down to the level where Mo once rubbed, Vaseline all over, one or two contacts, just to try to really throw this, this system out of whack, and see how does it, as we start to push it beyond what we'd see in a patient, does the system actually behave itself like what, Alex is getting? It doesn't do something so odd that, we get caught off guard. But the limit right now of our models is we're really kind of characterizing, I'd say, electrochemistry and electronics. We're still not pushing the envelope of how the brain might misbehave. And so, like one of my examples more in Alex's domain is, we published on this, it was in a sheep model. So that's the kind of the go-to also today, Steve, is either we go into animal models, which are expensive, and also in the 3R context, sometimes you're even more limited in a clinical trial. You just have such a finite space. You're not really pushing the envelope. It's more just like that check the box exercise as opposed to understand the envelope, the design envelope. But the hippocampus, it was looking at epilepsy networks. So you're like you start stimulating the hippocampus, and you get a nice little suppression of the activity, but then you go 100 microamps higher, tick on the knob, and you kick off an after discharge. And so it's this, the brain has these nonlinear properties, and then rebound effects. And that's the next stage. That's when we say, I'm trying to remember, like level 6 and 7 is where we want to go, because that's the next area where we can really take advantage of these models.
Right. Yeah, it's interesting.
And Alex with an X, what we found though is we prototyped as part of this little effort. John did what was from Tay Nedoff. We got to give Tay. We used Tay's epileptor model and prototyped it and put that in. So it's kind of a nonlinear model of excitatory inhibitory networks feeding back on each other, and it mimics some behavior. And it wasn't the regulators; they didn't even see it. It was the clinical partners we have. So that's not epilepsy. That's not what a seizure looks like. That's not the dynamic. And so I think part of it for us as well is not just to educate the regulators, but also to get confidence in the customers. The Alec Widges of the world have to trust these in-silico models if we're going to base any kind of conclusions on them.
At some level, cut ourselves a little bit of slack. I think the strategy for a long time was to build out these sensing and algorithm technologies alongside an established, kind of risk profile of deep brain stimulation, right? Deep brain stimulation was already on label. The research payload approach of basically always being able to piggyback off of an established therapy. You can go back at some level. There's a clinician programming the limits. So, and the patients can already change the device parameters within the limits. Well, all adaptive stimulation is doing then is just changing it in an automated way, what patients can already do today, right? That was the risk analysis that literally got us through into the first ADBS studies, was like, patients can do this today if they were automated and already changing the thing up and down. it's already kind of established as safe to adjust the stimulation in this sort of manner, right? And so, but all that work is based on, you know, decades of empirical evidence that led to the discovery of different targets that could be used for lesioning and then eventually electrical stimulation to treating these things. It's not like there is you know, some model that identified the STN as the target for deep brain stimulation. No, some poor sod got a brain injury there and his Parkinson's was cured, right? So at some level, but you know, the Parkinson's use case, I think in some ways it's almost a mirage just in how lucky we got. The place where we were sticking electrodes just happened to have a biomarker of medication and therapy state, right? Most of the time, the brain's not that simple. And while in the closed loop for Parkinson's case, it worked out because, you know, and we have this now wonderful adaptive therapy. Most of the time, you know, we're stimulating in places and we don't see necessarily an effect that checks with medication or behavioral state. And so I think we're at some point, we're kind of at the limit of what we can really do without understanding the brain. The empirical modes of algorithm discovery won't get us much farther, right? And I think that's one of the things is just to.
And on that topic, I mean, I feel like there's quite a, well, I don't know. The question is how much variability, and I think I know the answer, but how much variability do you have from patient to patient on this? And what does the learning process look like in terms of trying to understand the patients before you go and actually do the procedure to put these devices in? Is there a certain amount of information you can gather up front that helps in the calibration, the patient-specific calibration of the algorithms before?
Well, I'll tell you, in our stroke study, we have an exhaustive inclusion-exclusion criteria, which means that people have to undergo, you know, multiple hours of MRI to you know, evaluate specifically their cortical spinal tract. We do fMRI looking for motor responses. We also do TMS to look for an actual physical motor. And you know, at some point, we're already limited into, you know, ischemic stroke patients who are also not on anticoagulants. And I'll tell you, and then, you know, at some point, the tighter you make the net, the harder it is to find your unicorns who fit that criteria. And so, you know, it took us 9 months to find our first patient, just because, you know, at some point, the lack of understanding of how exactly all these criteria pieces need to be tuned and the lack of better understanding of what we're looking for leads us to then, you know, in some ways, just extrapolate old empirical measures forward and layer them on top of each other, try to find the right person. But, you know, this will help. I see Tim's raising his hand. He wants to get in here.
So I've got a question for you, Jerry, because it's to Stephen's point. So like one of the nice things, benefits of a lot of these devices, you get some reasonable performance for open loop. So when it comes to developing, with the focus of the In-Silico being on these adaptive methods and testing them, you get some marginal benefit from open loop. I think that's also the case in your application, Jeff.
100%. 100%. Every study is our open loop. That's what we start with, and like we can always fall back.
Exactly. Yeah, so that's same in cadet, where the initial trial is actually open loop stimulation. And then to your point, Stephen, then we also get the opportunity to characterize the signals, the physiologic signals, and even in that patient, there's specific artifacts and how it couples.
Yeah.
And then what we do is we pull that data set. So Tori and John, part of their job is they characterize during the trial, pull the data set in, then we build this kind of hybrid In-Silico model. Then we can test out the proposed algorithm on the bench and say, yep, it actually looks like it's going to work okay from in terms of the safety elements, like what Christine is in from MHRA. So we can say to Christine, well, it doesn't misbehave from the standpoint of the device and the interface. Now we can load those parameters, you know, through the wireless radios, and same in Jeffrey's, the wireless radio updates. And then we test, but what's missing is that last step of we still require the clinical neuroscience then to run its course. You know, we can't make predictions on the therapy. It's more just about the safety at this time.
Yeah, man, that's hard. Mark's got, yeah, let's hear what Mark's has to say.
Hey, I really appreciate the presentation. Still trying to take it all in. A lot to learn today, which is great. But I think the thing that, and echoing one of Alex's questions is, what models are sitting inside? So when we think about this project, and the CEiRSI project overall, we all recognize that modeling and simulation is being used as part of medical device development. But then the question is, how can we enable those models to be accepted as part of a regulatory submission? So then that brings us to this whole credibility assessment. So I know I saw your question of interest and context of use and risk, but I'm wondering how do we apply that risk? Like what is the model? So you mentioned one on I think the neural response, and then another one around the modeling of the brain overall, if I remember correctly, or brain activity. So when I think about this project, I think we focus on those types of models, physiology, physics, etc. So I'd wonder, how do we establish the credibility of those inside of your control algorithm? I assume those models are activated, and those are what help the device make a decision about what to do next in terms of stimulation. Is that true?
Tim, you're muted.
Talking to Tori, we're going to pull up a slide. There's a proposal for Mark. Like one next step in the area of the DBS as it exists today and looking at Parkinson's disease. We're just going to pull back up the transfer function. We have a colleague here, John DeBarros, who works with Wei-Ling Tan. And what they're doing is that model, the staircase one.
Oh,
Yeah, so that staircase step of stimulation going up, which we'll share here in a sec. You're just doing this to get a new laptop. So there we go.
There's a delay too.
Yeah, okay, so no, go
Not
up.
this one?
No, not that one. Yeah, that one. No, I'm sorry, that one will do. Sorry. Yeah, so on the right, panel B. Yeah, so panel B, you can kind of see, this is from Jeffrey, kind of his collaboration with Helen Bronte Stewart at Stanford. As you can see, there's this stimulation transition of, here's our amplitude going up, up, up in the middle row. There's the beta power on the top and how the brain's responding, and then also some symptoms. And actually, in this case, as I say, we're playing basketball, so higher is better. This is measuring your ability to move your hands. So it's Parkinson's, so bradykinesia, slow movement or lack of akinesia, lack of movement is a symptom. So you can see as you go higher, the beta power goes down and your clinical symptoms improve. And what we want to do is mimic this as a first order proxy where we can have kind of the nominal patients and then a few outliers and have basically put this in, and then we can actually provide stimulation outputs from the device and then mimic these physiological signals on the top row and then use that in a closed loop manner. And I think with that, it's kind of it won't have emergent properties per se, but it will at least allow you to vet through kind of a library of different signals that we might run into testing out a new device on a de novo, Parkinson's patient. You'll at least be able to bound the response of the device.
So it's a database model that you'd be using to tune the performance of the device to each patient. I mean, I guess I'm wondering is, okay, so it'd be purely a database model that you're using, and that model is fine-tuned and retuned for every individual patient, based on, what's happening in the clinic. There is no baseline model hiding inside. So I'm trying to think of an example of, what's a good example, guys? Something where you have a model that you run on a specific patient. We have these for like stent deployment, where you do a surgical planning for a stent to a specific patient based on MRI data that you take. You say, this looks like the right size. We simulate the delivery and the implantation, and we say, yeah, let's do it, right? And in that case, the physics framework is the same underneath, but it's all driven by physics. So software is a medical device type application. So, but it's, more mechanistic or physics based. So what you have is, I guess, a SAM-D that's more data-based? No
For
physics?
now, yeah, for right now, it's more database. The there are some mathematical models that are being developed.
Right.
And in terms of nonlinear oscillators, the Wilson-Cowan framework and the like, where we can start to build that and start to apply it. And that would be the next step. But I think this is fair to say within our team, and Alec and Jeffrey, or John, Tori, cut me off if you don't agree. We also need to ease the field into it. So I think that's one, starting out with a database model, and then building from that is a way to actually give the community, both our clinical partners as well as our regulatory partners, confidence in what we're doing. And so I think, Alex F, I think within our domain, I think we were always one of the work plans that was out there. Like we were kind of the oddball of trying to figure out how to do in silico brain models. And then part of what we're trying to do here is map a continuum of stuff that really is not that glamorous, but actually has meaningful short-term results. And we use that when we use this hybrid in silico to show the devices behave themselves in a neuro closed loop environment, and we show that worked, then we start to build the next section of the bridge to a database model to predict how the device should respond and check that it does what it's supposed to. And then ultimately getting to this, I'll call it, like you say, Mark, a physics-based, neurophysics-based model that gives us more emergent properties, which is what we all want to aspire to. Because that's what Alec really needs for some of his exploration, is to test out a true neuro algorithm model before it commits to a clinical study.
Right. And a way to say this is, Tim's right. Database is a place you start. Eventually you build from that to, here is what's not contained in the database, but on the basis of what is in the database is a reasonable extrapolation, especially because we've done a sensitivity analysis to show that small changes to the assumptions do not wildly lead you off into cones of differing outcomes.
Mm-hmm.
One big part of that, though, is I think the hard part is the building of the database, right?
Also
At some
true.
point, you don't have a good database for what's the beta response of different stimulation amplitudes in the brain until you kind of already have identified that the beta is something to be paying attention to, and we should be collecting these experiments to collect this data. At some point, when we're getting to the adaptive territory, though, too, it's like oftentimes the data that's needed is trial runs and feasibility runs of algorithms in ways to collect data to support modeling. So you can't just use like old static open loop data to work your way into a dynamical model of a system in some ways. And that's where I think at some point, when we're seeing the efforts around, we need to be supporting, I think, a lot of the early feasibility work, collecting data with, in limited patients. And then, clearly all that data needs to be archived and shareable and managed in a way to allow them to further discovery. Because at some point, you can't really, does need to be some space, I think, for people to go out and try the crazy things and collect the data to then enable the modeling, because someone has to probe the system before you can actually start to model it, right? It's just how do we do that in a safe way with the right devices is the trick, I suppose.
I think I should have attended more of your meetings. That's my conclusion, or any of your meetings. I'm not sure I got to attend any of them. But anyway, yeah, I have other questions now, but Alex has patiently had his hand up for a while, and we're running out of time, so I'll let the boss go last. Yeah, go ahead, Alex.
Yeah, so I two things. This is really important piece of work, Tim, because I think we need to be careful as a community that we don't end up boxing every technology with the same sort of credibility assessment framework that we have. That may work well for a broad set of technologies, but not for all of them. And this stress testing with approaches that we haven't considered so far, I think is really helpful. I do have some suggestions that I will make offline about how you could actually go from what we were discussing, a database approach, to actually a data-driven model. It still is data-driven, but at least it's a model, and there are credibility frameworks that we have explored for some of the other pilots in this particular space. So I'll we'll discuss this offline just to avoid enlarging here. But I wanted to ask you one question. Can you comment a bit how these approaches and everything we've been discussing is different or is similar to what you would do for other systems, like the insulin pumps type of example with closed loops that you mentioned before? I was saying here that the approach would be, if you think of this from a credibility point of view, whatever methodologies we develop for one would equally be applied to the other one. It's completely different models and different loop systems and different sensors and different actuators, but otherwise the approach would be similar. Or do you think there is something fundamentally different between these two? And second, I suspect that there is more maturity in some of these other systems than in the brain ones, due to the brain element, which is a lot more complex to understand, the basic science. But I don't know if I'm completely off the mark there. Could you comment? Well, any of you, could you comment on those 2 points?
Okay, maybe. Hey, Alec, do you want to? I think the thing that comes to my mind is just the diversity of the brain networks compared to some of the more contained organ systems. So like in cardiac, we have very specific subdomains of disease that are fairly well characterized, and we can reduce those to a fairly finite set of modeling equations and explain what we expect to see. Like we say, epilepsy is a catch-all term. Many things going on in neural networks, and I think it's that some of that heterogeneity we have to deal with. But I'd say you're kind of that expert on the neurophys challenge of the brain.
Well, and I think, in some sense, right, like, the Alex's questions D and E in the chat are kind of both related to what you just said, and both part of the answer here, right? Because what you're highlighting, Tim, is we can't just talk about the brain, right? What we are talking about are and we may not even be able to talk about a given illness. Go back to Parkinson's. Within Parkinson's, there's one algorithm that you may want to choose with one time constant for what we somewhat understand, which is suppress tremor by suppressing beta van, the example you show. But consider freezing of gait, which is an episodic event triggered by environmental stimuli and has a totally different biomarker with a much more stochastic time frame. Consider what we're increasingly learning about sleep and cognitive disturbances, the canonical non-motor symptoms that, for example, leverage some of the circadian rhythms that you were showing and you've written about in some of your other work. The same thing is true when we consider the psychiatric space, where there are some people who are interested in suppressing a momentary paroxysmal symptom, a surge of anxiety and post-traumatic stress disorder, an immediate urgency in obsessive compulsive disorder. And there are other folks who are interested in smoothing out the very long-term, almost ultradian phenomenon that is mood variation. Those are all different things, different controllers. And so what this highlights, though, this gets to point E, Alex, is how mature is the underlying science? In many cases, not, because I forget who made the analogy that there were too many things we learned in drug development that we couldn't do in mice because their livers are designed for a different thing than the human liver is designed for, right? We're talking about an animal that eats rotting garbage. It has a different chemistry than people. And as such, there are things you can only do in people. So the question is not, are you ever going to have a mature model of how the human brain is going to respond to a proposed therapeutic? You are not. You cannot, without in fact doing the experimental device investigation. What you can do is sandbox and say, for a reason, and I'm gonna just keep coming back to this every time you call on me, is what we're talking about here is saying, for a reasonable range of assumptions, no catastrophically bad outcome is likely to occur within a reasonable tail of a probability distribution, which is how, when we all know this, That's how you do risk management. You never guarantee, you don't have, aside from simple linear dynamical systems, which don't exist in nature, you don't get guarantees. You get probabilistically safe enough outcomes with good capacity to detect deviations and rapidly go to fail-safe mode and investigate cause of error. That's everything. That's everything we're doing here.
Can I just ask you a final question that was raised by Michael? I reckon that we are on the time, and I would like to suggest that when you wrap up, Tim, the pilot into your sort of mini report that comes at the end, you might want to explore some of these questions that we haven't had the time to go through, because, you know, in a sense, this helps us to understand what needs to be done on the credibility sort of framework point of view to encompass and bring these sort of technologies as well. But I want to come to Michael McCrell question. So I think if I understood correctly his question, he would like to understand what is the worst thing that can happen if things go wrong and you get this the sort of stimulation limits out of range. I suppose thinking in the framework that has decision consequence and, you know, a model impact as well. So what can happen? It's just discomfort, is actually genuine life-threatening. Can you know, what happens in those cases?
One risk we control in stroke, as an example, is that motor cortex stimulation above motor threshold can cause seizures. That's an example. So don't wrong, we do a bunch of clinical observation of tuning of stimulation parameters to make sure we're not going to do that. But if the system was to be completely uncontrolled and unpredictable, you know, inducing a seizure in a patient is pretty close to one of the worst things you could do. In the more common cases in deep brain stimulation, you can cause everything from tingling sensations, which are relatively harmless, to eye closure, to difficulty speaking. But imagine, you know, if someone was driving down the road and, you know, the algorithm didn't behave the way it was, or created, you know, some eye closure event or something like that. You know, like, yeah, you can start rapidly kind of curving into areas where some of these things would be difficult to manage. The likelihood of that actually being something that a clinician would like enable or set up, you know, no, those would be side effects that they would be avoiding.
Well, so that Jeffrey, is the earlier point on we can end up and move into sort of getting severe dyskinesias.
Yeah.
Or the other, so you're kind of unintended motion, but also, Alex, when like the FDA guidance is unintended cessation of stimulation can lead to, like Alec was saying, freezing a gait, a fall, which a fall can actually be lethal in these populations. And so there can actually be pretty severe side effects. And so that's why one of our takeaway messages, not something that, you know, to close on, is we have the existing standards like 60601-1-10 help to define safe operating zones and boundary limits that also we're applying today, even in the context of a manual closed loop, because patients oftentimes have their own controller, but they can make adjustments. And so the Alex of the world, Alec Widges, will actually set boundary zones that they have to stay within. But there can be very severe consequences, including life-threatening ones, if these algorithms are not designed in a safe manner.
Okay, and one last very quick question. That's your community. By community, the people working in neurostimulation of some sort. Are you familiar with were you familiar before this exercise with the ASME BMB40 standard? Is that something that is being used already, or it has been for you like something new?
That's new.
Okay, that's interesting. And
yeah, at least as far as within our domain, the ASTM standards and the modeling, that's a new, like the credibility framework and the evidence. Yeah, that's something that Jeffrey and Alec and I are socializing within our community.
So can I suggest, because this also is an interesting, for us, it's an interesting experience in the sense that there are, I'm sure, many more device domains than these ones that have not been exposed to this way of thinking of the ASME BMB40 standard that comes more from a mechanical engineering community than an electrical engineering community. Now, there is now a development of an international version of the ASPE BMB40 by ISO. And team, there's a way for, I can tell you how to do it, but it's a way for people from the UK to be nominated to be members of that standard. And I'm sure there is another way to do it from the US or any other geography, but you need to look at what are your nominating bodies. I can tell team how to do it in the UK. It would be great to have someone from your community participating of those to make sure that, you know, whatever comes as, this is going to be credibility standard for the use of modeling and simulation in medical devices. So in principle, any type. And just to make sure that these would fit to this particular sort of neurotech community. And yeah, and that hopefully is something that could be helpful. Well, let me just say a big thanks to the whole team, to everyone, for all the time you put in preparing this. Was really, really enjoyable. We learned a lot. As Mark and Stephen say, probably a lot of things a bit about our you know, went above our head and our experience, but it was really, really helpful. And I wish you a great rest of the day to everybody, and look forward to continue the conversation. Thanks, everyone.